wish your doctor had a tricorder like they had in Star Trek? You know, the one they pointed at you and it figured out your illness and what the treatment should be? Well, that day is not far off. In this, our final lecture, let's examine the exciting discoveries and technologies that are already creating a world that was only recently in the realm of sci-fi. With the knowledge you have gained, you will also be able to follow exciting developments that are in the pipeline that may one day make tricorders as outdated as rotary dial telephones. We already have smartphone-sized diagnostic devices that are smaller than the tricorder. Our devices can detect TB, malaria, and a range of other communicable diseases. It can tell you the exact genetic strain of the pathogen, and it can even provide treatment recommendations. Designed for use in developing countries, these tiny marvels were made possible through the convergence of advanced engineering and genomics. And it can just as easily be used to detect cancer or genetic diseases, all in your doctor's office. Devices like it will soon replace expensive and time-consuming lab tests that made you wait for weeks for an answer. And it will increase the likelihood that the treatment you receive will be effective. A device like this is possible because of discoveries made by molecular biologists who study the weirdly named omics. Omics are the leading edge of an emerging new approach to medical care, sometimes called P4 medicine. An approach that is predictive, preventive, personalized, and participatory. It contrasts with traditional care, which is mostly about reacting to problems and treating symptoms. The arrival of omics started with genomics, which was itself spawned by the completion of the Human Genome Project in 2003. That massive undertaking determined the order or sequence of every single base in the human genome. One of the benefits of this project was the development of ways to sequence DNA that are 1,000,000 times cheaper and 5,000 times faster, to the point that today you can get your entire genome read in about a day. And in this case, it would be your specific genome with its own tiny quirks. Now, of course, this is a lot of information to have about yourself. Theoretically, storing 3 billion base pairs of information would take up most of a good-sized thumb drive, depending upon how much peripheral information you included. Moreover, accurate sequencing is not a one-and-done sort of process. The quality of your data strongly depends, for example, on the number of times each base in the genome is read during sequencing. A sequence that has not been read and checked multiple times is called a draft. Fortunately, costs have fallen dramatically. Starting in 2001, researchers had called for the cost of sequencing a human genome to decline to less than $1,000, less than an MRI at some hospitals. That goal was achieved around 2014 to 2015. The next goal is whole genome sequencing in about an hour for $100. There are even small, sleek, portable sequencers that look like a little flip phone and fit in your pocket in case you have a sudden sequencing emergency. Genome sequencing is not just for humans. The entire DNA sequences for thousands of other organisms have also been obtained. There are now huge databases filled with sequence information generated by these projects, and more information is added each day. Side by side with getting genome sequence data, scientists are also determining and cataloging the structure of thousands of proteins. The easy availability of so much genome data allows for a whole different level of analysis called genomics. Entire new fields of study have sprung up to analyze the flood of data. For example, bioinformatics and systems biology. Besides genomics for DNA, there are several other omics. Transcriptomics for RNA, proteomics for proteins, metabolomics for the metabolites in a cell. These fields aim to look at all the genes, all the RNAs, all the proteins, and all the metabolites in a cell simultaneously in order to obtain a complete picture of what's going on and when. These are questions on a grand scale. For genomics, researchers are examining DNA sequences, not just to determine protein coding locations, but also the functions of encoded proteins and how they might be regulated. With information from so many species available, comparative genomics also becomes possible, where processes central to evolution may be revealed. Central to understanding information in DNA is genome annotation. The sequence of a genome is just a string of billions of bases. Scientists use algorithms that look for features such as protein coding regions, potential regulatory elements, and so on, so that function can be assigned to each sequence in the genome. This gets duly noted in the form of a note or annotation that helps other researchers to use the information properly. All the genome, RNA, and protein sequence information and annotations are stored in databases across the world. While the data can be accessed by anyone with an internet connection, the raw information is pretty incomprehensible without specialized knowledge and understanding. At some point in the future, friendlier interfaces may permit non-experts to interact more meaningfully with the data. Now, researchers are steadily identifying the variations in the DNA sequences of different individuals and the significance of these differences. These patterns of differences can be used in the diagnosis and personalized treatment of disease. Normal cells and cancer cells can be compared to determine which proteins are involved, how those proteins interact with each other, and what cellular processes are altered. Meanwhile, genome analysis is adding to our understanding of biochemistry as an integrated whole. And the study of the small genetic variations among humans has already provided quick returns. Only about 0.1% of our genomes varies from one human to another. But those small, subtle differences can have consequences for our health. They can affect whether and to what extent we get sick, and even how we respond to treatments. Some differences are in protein coding regions, so they affect the function of the encoded protein. And you already know that even the mutation of a single base can have negative implications for health. For example, scientists have long been curious about people who have unusually low levels of LDL cholesterol in their blood. You know, it's that great uncle who ate fried eggs and bacon for breakfast, a couple cheeseburgers for lunch, and steak and gravy for dinner every day, and is still boogieing around at 99. No matter what they do, these folks don't ever develop high cholesterol levels and seem to be protected from cardiovascular disease, while leading a life that would long ago have finished off the rest of us. What's the secret in their genes? Genome comparisons of their sequences with those of the general population showed that they have a mutation that knocks out an enzyme called PCSK9. Now, PCSK9 provides normal breakdown of LDL receptors on the surface of cells. Those receptors capture LDLs from the blood and bring them into the cell. When the enzyme is knocked out, more LDL receptors remain on the surface of cells. So people with the inactive PCSK9 enzyme had even more LDL receptors than normal on their surfaces. So, cells of people with this mutation remain better at taking up LDL cholesterol from their blood, and their blood cholesterol levels were correspondingly lower. On the other hand, for those of us with normal functioning PCSK9 enzymes, we have fewer LDL receptors on our cell surfaces, which leaves more LDLs in the bloodstream. However, the genomic comparison is also useful for those of us who aren't so lucky. Researchers designed new drugs to inhibit the enzyme from breaking down LDL receptors. 2 drugs to inhibit the enzyme were FDA approved in 2015 under the names alirocumab and evolocumab. The new class of drugs discovered through genomics provides an alternative to statin drugs used for high cholesterol levels. Scientists are also using cataloging single nucleotide polymorphisms, or SNPs. These are specific places in genome sequences where a single nucleotide differs between groups of people. Say an A is replaced by a C. An SNP is a point mutation, but one that is found in many individuals, not just one person. SNPs are dotted all over the genome, about once in 1,000 nucleotides on average. Of the millions of SNPs in our genomes, many are not in protein coding regions, and a single SNP is not very informative. But researchers have done what is called genome-wide association studies, or GWAS, that look across the genome to see whether some SNPs are more prevalent in people with a particular disease compared to those who are healthy. Such studies have shown that some patterns of SNPs in a stretch of DNA are found to be associated with susceptibility to a given disease or response to a medication. This could be because variations in SNPs might change how gene expression is regulated, for example. You can think of the pattern of SNPs like a barcode. A particular combination of SNPs inherited together from one parent, or that tend to be inherited together across many people, is known as a haplotype. A person gets half of her chromosomes from mom and the other half from dad, so her full SNP profile will have two haplotypes. Mom might have two different haplotypes to offer, while both of dad's haplotypes might be the same. Whatever the full SNP profile is could make one child more likely to benefit or to have an adverse drug reaction from a particular treatment than someone with a different SNP profile. For example, statin drugs are often prescribed to manage cholesterol levels. According to the CDC, 43 million Americans took statins in 2015. While they are generally well tolerated for some patients, statins cause severe muscle damage or myopathy. Genome-wide studies have identified specific SNP patterns that greatly increase the chance that an individual will have this reaction to statins. This information can be used to avoid prescribing statins to patients who might be harmed by these side effects. SNP profiles can also help in decisions about whether an individual will benefit from the asthma drug albuterol, and in determining the dosage of the blood thinner warfarin for a particular patient. The use of such genomic profiles in deciding how to treat patients is called pharmacogenomics. The FDA has begun to provide information based on this knowledge, and in some cases recommends patient testing before the drug is prescribed. For example, before prescribing abacavir, an antiviral drug, the FDA recommends that people be screened for a particular variant of a gene called HLA-B. Individuals with this version of the HLA-B gene are high risk of having a severe allergic reaction to the drug. With more genomic information available, doctors will be able to choose medicines best suited to each patient, rather than a one-size-fits-all drug. For some conditions like cardiovascular disease, inflammatory bowel disease, and breast cancer, genome-wide studies of SNPs have been very useful in risk prediction. For other multifactorial diseases, SNP profiles are not yet sufficient to provide clear-cut answers. This is partly a function of the complexity of the diseases. There are often many genetic factors which interact with each other and with the environment. Scientists are still determining what all those pieces are. Because of this, people who send away to companies offering personal SNP profiles or sequencing of their genomes should be aware that information is not available to provide a complete picture of their disease risk. Of course, cataloging of small variations in the genomes of individuals is also useful for tracing human migrations and ancestry. The patterns of variation used in such studies focus on mitochondrial DNA to focus on the maternal line and Y chromosomes to trace the paternal line. The DNA in our mitochondria is inherited from our mothers. This is because in the production of a fertilized egg, the egg is usually the source of mitochondria. Except in rare cases, sperm do not contribute mitochondria. This means we inherit our mitochondrial DNA from our mothers who got it from their mothers and so on back down the female line. Likewise, the Y chromosome is passed down from father to son down the male line. In lines from both parents, mutations in the DNA are passed on, and sets of common SNPs can be used to trace a maternal or paternal line of inheritance. In addition to SNPs, Y chromosome analyses also include variations in the number of short repeated sequences called STRs. These are short repeated sequences present in varying numbers in different individuals. For example, one person might have 12 repeats of the sequence, another might have 16. The numbers of these repeats, like the SNPs, are inherited by sons through the Y chromosomes. Just as we saw before, particular combinations of variations are used to define what are called haplogroups, groups of people with a shared haplotype. If you share the same mitochondrial haplogroup as someone else, you share a maternal ancestor somewhere along the line. Likewise, if you're in the same Y chromosome haplogroup as another person, you have a male ancestor in common with that person. Still, it's important to remember that these haplogroups represent ancient branches of the human family tree going back tens of thousands of years. Everyone in a given haplogroup can trace their ancestry back to a single individual in the deep past. It's entirely possible to, for example, be from Korea and fall in the same mitochondrial haplogroup as someone who was Native American. People migrated across the land bridge from Asia to North America in the past. So both Koreans and the descendants of those who migrated from Asia to North America could have had a common female ancestor who lived in Asia a very long time ago. On the Y chromosome, an eye-popping example is the legacy of Genghis Khan, the founder of the Mongol Empire who lived almost 1,000 years ago. Today, Genghis Khan's Y chromosome haplotype is seen in over 16 million men. Genghis Khan is said to have fathered over 100 children, and of these, all of his sons would have inherited his Y chromosome. But his sons in turn must also have been prolific, passing on the legendary chromosome to their numerous sons. So far, we've only looked at sequence information in DNA and how that can guide decisions in medicine and be used to trace deep ancestry. But as far as life is concerned, variations in DNA are only important if they're expressed in some way. Other omics look at the actual use of the information in DNA, that is, which genes are transcribed and what proteins are made. Transcriptomics analyzes transcripts, that is, RNAs, on a genome-wide level. This allows us to know exactly which genes are being copied into RNA under different conditions. For example, it is possible to identify all the genes transcribed in response to the treatment of cells with a hormone, or to compare the global transcription patterns in normal and cancerous cells. This is made possible by development of technologies for rapid sequencing of thousands of RNAs. A technique called RNA-seq is used to detect and measure the levels of every transcript in a given sample. Transcriptome sequencing can provide information about all types of RNAs, including regulatory RNAs like microRNAs. So sequencing of the regulatory RNAs can tell us about when certain genes might be silenced, while sequencing of the other RNAs provides a complete profile of all the protein coding genes that are expressed. For example, transcriptomics has shown that in some cancer cells, regulatory RNAs are made that shut off production of tumor suppressor proteins, allowing cancer cells to proliferate unchecked. Let's think about what this means for medical care. Getting your genome sequenced, if you want to understand what's happening with many ordinary ailments, such as a cold or pneumonia, say, will have almost no meaning. Your genome doesn't change during an illness. But the transcriptome changes in response to everything. Knowing how the transcriptome changes in response to drugs, infection, cancer, and so on is going to be a treasure trove of information for better diagnosis and treatment. Besides studying genomes and transcriptomes, researchers also analyze complete collections of proteins with proteomics. All the proteins that are in a particular cell type or tissue can be extracted, separated, and identified. Differences in protein profiles between normal and cancerous cells may provide clues to the mechanisms by which the cancer arose or suggest ways to treat the cancer. For example, normal cells in a sample may be making the proteins encoded by genes A and C, but not B. By comparison, the cancer cells may be making large amounts of the protein encoded by B, but none of the protein encoded by gene A. The cancer cells may be making the protein encoded by gene C, but perhaps it is a mutant form. This information is useful in understanding the underlying causes of cellular dysregulation. Lipids are another group of molecules that give us insights into many medical conditions. Advances in the techniques for studying global lipid profiles have opened up the field of lipidomics. Information about how lipids vary in cells under different conditions help in predicting the risk of metabolic diseases like diabetes, non-alcoholic fatty liver, and cardiovascular disease. Lipidomics can also show us how diets affect cellular lipid profiles. Lipid profile changes can also tell us how effective drugs are. So for example, glucocorticoid drugs have been the standard treatment for a lung disease called idiopathic pulmonary fibrosis, or IPF, where idiopathic means the cause is unknown. Patients with idiopathic pulmonary fibrosis have changes in lipids in their livers that can be tracked using lipidomics. People diagnosed with this disease survive for only 4 to 5 years, even with the treatment. Finding alternative treatments is a high priority. One candidate treatment was a drug called Biochemistry. To test whether the drug would be effective, researchers treated mice with IPF. And because the new lipidomic methods were available, they also compared the global lipid profiles of mice with IPF that were not treated with the drug and those that were given the standard drug. Not only did Biochemistry appear to cure the lung disease, the treated mice had liver lipid profiles similar to healthy mice. Animals given the standard treatment, on the other hand, had little improvement in their lung symptoms, and their liver lipids were more out of whack than mice with the disease that received no treatment. Lipidomic studies like these can help screen out treatments that have unexpected and severe side effects and identify those that normalize biochemical profiles. Lipidomics is complemented by global measures of molecules like carbohydrates and other classes of cellular molecules. All these are grouped together in a study of metabolites known as metabolomics. Together, these fields are allowing biochemists to have, for the first time, a big picture view of the activities of cells. Integration of information from the genome, transcriptome, proteome, and metabolome will show how these components are interconnected and how these interactions determine biological functions. Some analyses add the interactome that defines interactions among proteins. But researchers don't stop at a snapshot of what is happening in cells at any one moment. Cells are not static entities. Every moment, cells adapt their activities in response to changing internal and external conditions. To understand these complex systems as an integrated whole, systems biologists are developing models that depict these dynamic interactions within cells. The goal is to explain how interactions within cells give rise to the properties and behavior seen in disease or health. These interactions can be represented as networks that influence each other to keep everything running smoothly. Disease, then, can be thought of as anything that perturbs these networks and necessarily has wide-ranging effects. Systems thinking and the integration of vast amounts of information about molecular events and processes is changing the face of medicine. As the omics allow us to understand risk factors better, we can predict who is most likely to develop certain diseases. For many conditions, this can lead to preventative measures that can help head off or reduce the risk of actually developing the disease. This approach favors the maintenance of good health or wellness rather than the current approach of fixing each problem as it arises. And in case you're wondering, the Genetic Information Non-Discrimination Act of 2008 protects Americans from discrimination in health insurance and employment based on their genetic information. The availability of rapid whole genome sequencing and immense amounts of reference data enables the personalization of not only recommendations for maintaining optimal health, but also treatment when needed. Individual whole genome sequencing is neither necessary nor routine for most situations. But to get a sense for cases where it is invaluable for diagnosis and treatment, let's look at an early success story. In 2011, a striking case that used whole genome sequencing to solve a medical mystery was reported in Science Translational Medicine. The patients were twins, a boy and a girl, who had been misdiagnosed at age 2 as having cerebral palsy. By the time the kids were 5, it became clear that the initial diagnosis was wrong. They were then diagnosed as having Sagawa's dystonia, a condition where the brain makes too little of the neurotransmitter dopamine. The treatment for this deficiency is L-DOPA, the same drug that's used for Parkinson's disease, another condition in which too little dopamine is made. This seemed to help some, but as the kids got older, more problems surfaced that L-DOPA apparently couldn't fix. It was at that point that their genomes were sequenced. With the full genomic sequence, researchers were able to pinpoint the source of the problem. And it turned out the twins didn't just have a problem making dopamine. They had a rare mutation that made them unable to make another neurotransmitter, serotonin, as well. With this information, doctors were able to prescribe 5-hydroxytryptophan, a molecule which is converted into serotonin. And boom, just like that, the kids improved. They went on to play sports and have a normal, active life, just like their friends. A mere 8 years after the completion of the Human Genome Project, 2 kids have their lives back and their health back, thanks to genome sequencing. While we focus so far on medical applications of the omics, they have already begun to change the world in other ways as well. Having the complete sequence for crop plants and livestock makes it easy to identify individuals with the most desirable traits. DNA markers for drought or heat tolerance in crop plants will be useful for developing varieties that can thrive despite climate change. Dutch researchers studying genetic variations have identified cows that naturally produce low-fat milk, no skimming needed. These animals may then be used for breeding, or the traits may be transferred by genome editing. DNA sequences can also be used in tracking food products. It becomes possible to determine the source of milk used in making a cheese. DNA analysis can also be used to tell whether the beef has been adulterated with cheaper horse meat, and to determine that meat sold as kosher or halal does not contain traces of pork. It's also used to detect poaching of protected species of animals. So the omics are here to stay. They are also one of the fastest moving areas of life science research. They've taken us from the zoomed-in study of single genes and their products to a mind-blowing view that is the molecular equivalent of seeing the whole world from outer space. This remarkable combination will be bringing us amazing new advances for the foreseeable future and remaking the world in ways we never imagined. I hope the knowledge you've amassed in this course leads not only to a better understanding of the links between molecules and life, but also to a new avenue for lifelong curiosity. The root of all scientific investigation is ultimately curiosity. From Isaac Newton's apple tree that led him to the discovery of gravity, to NASA scientists designing instruments to detect the biochemical signatures of life on other planets, curiosity is the engine of scientific advancement. One of my joys in teaching the subject of biochemistry is fueling the curiosity engines of my students. Some describe teaching as a noble endeavor. I view it as a privilege, and I'm honored to have had these sessions with you. As for your curiosity, let it go forth and multiply. I close with a verse. So now we all have reached a point where the background's been explained. The proteins, fats, and carbs in cells, how enzymes get restrained. How pathways run and building blocks make biopolymers. The world of biochemistry is now becoming yours.